KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, Xylem, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video, so we are discussing dicotyledonous leaf. So in the side, so we will discuss the entire information related to the dicot leaf. It means plants, most of the plants we will see, no. So all those plants are dicotyledons, and monocotyledons also we will see. So I am not objecting. Now in this video, so we are discussing both dicot plants and monocot plants. Monocot plants leaf on the side is there. So both dicot plant leaf as well as monocot plant leaf also both we will discuss in this video. So let us start to discuss this particular anatomical or anatomy of dicot leaf and monocot leaf respectively. Now here let me explain why this particular dicot leaf we are considering by one more name, or we are calling by one more name as dorsiventral. Once you see there, dorsiventral, ipo zoology point of view, dorsal side and ventral side, I think so you have heard this one. Means dorsal side is there, even for our human beings also dorsal side will be there as well as ventral side also is there, means towards the stomach and towards the backbone. So two places, likewise how we will be having, no. So likewise dorsal side and ventral side also we can see in one particular leaf. Okay, so that's why here both are differentiated. Both are there, and suppose if both are differentiated means, so such particular leaf we will be taking as dorsiventral leaf. Try to learn. Ipo example paringa, if you take one dicot leaf, no, if you take into your hand, no, if you observe that one particular leaf, upper surface and lower surface, no, color thickness will be different. Did you notice that? So means sir, why sir here, why sir color differentiation is there, why sir upper side no romba thickness irukku but lower side no koncham pale color is there, why sir means, so while explanation is going on I will tell you that. But here what is the point, dorsiventral leaf means dorsal side as well as ventral side both are differentiated. Both are differentiated. Next, now here vertical section. Here leaf point of view, what is the section we will do means vertical section we will do. So simply this vertical section, so we can take it as VS. V dot S. VS section we will call in case of leaf. Vertical section only we will take. Suppose in case of roots or in case of stems, no, so we will be taking transfer section. Listen, carefully try to remember. Roots or stems, if you are taking into your hand for cutting, no, we will do transfer section. Suppose leaf you are taking into your hand for study of anatomical structure or for study of anatomy, no. So there, what is the section we have to do? Vertical section we have to do. I hope you understood this one. Now here, what is the main important point, or what are the points we have to discuss, no? So all important points I have made as like underlines also. Okay, now let us learn peacefully, and while class is going on, maximum members try to learn the information easy da. Okay, even a neat bits also I will cover while going on smoothly. Now there if you take one dorsiventral leaf through the lamina, so three main parts. So definitely we can find out the presence of three main parts, namely what are the main parts. So we can see in a dorsiventral leaf means namely epidermis, mesophyll and vascular system. So first of all epidermis we will observe, later mesophyll tissue we will observe, and finally vascular system also we will find out. Okay, now let us see information of epidermis and information of mesophyll and information of vascular system also. Now let us see. Now the epidermis, first one epidermis which covers both upper surface. Once you see epidermis which covers both the upper surface and lower surface. Both here we have written, that's why we have to take two. One is upper surface and one more is lower surface. Now upper surface, our subject point of view, we are calling as adaxial surface. Adaxial, don't forget adaxial, ADA. Adaxial na upper epidermis. Appuram lower epidermis na abaxial, ABA. If ABA comes means lower epidermis, ADA comes means upper epidermis. Simple the once if you consider means definitely you can save this point in your mind. So upper epidermis we will be calling by another name adaxial, and lower epidermis we are calling by one more name abaxial. Of the leaf has, now here once you see there one by one, the leaf means here this particular leaf has conspicuous cuticle, means here the both epidermis is covered by which one? Definitely we can see the presence of conspicuous cuticle, means here the presence of the cuticle very clearly we can understand or very clearly we can observe. Means here if suppose listen one small example, do you know water lily? So water lily or like chinna chinna hydrophytes are there, like a lotus flower producing a plant, did you see? Lotus plant. So there on the leaf, no, suppose if you put one drop of the water, no, how it will move, that particular one drop of the water, it will move like which one, mercury only, no, so chemical, so that one like here also. Why sir here that particular leaf means what is a water drop we are keeping on the leaf, no, that one will not be like at one place, that will move. Why? Because there on the leaf surface, no, we will be observing waxy coat. We see no waxy coat like structure which is present on the epidermis of leaf, we will be taking as a cuticle, we don't know. So this particular cuticle only helps in reducing of rate of transpiration. Already in tissue system, in epidermal tissue system we learned. So likewise here also epidermis both sides are definitely covered by conspicuous cuticle. So definitely leaf epidermis is covered by cuticle layer or cuticle. So now abaxial epidermis means now other talking about which one, either lower or upper. Lower. Why? Because here abaxial abaxial epidermis generally bear more number of stomata or more stomata than the adaxial epidermis. This is neat bit. Now, simple information point of view, lower epidermis, let us take like that. Lower epidermis generally bear more stomata than the adaxial, means upper epidermis. Means in comparison of lower epidermis and upper epidermis. Listen carefully. In comparison of upper epidermis and lower epidermis, so where we can find out the presence of more number of stomata, we can find out the presence of more number of stomata on lower surface. Lower epidermis. So thereby here means on the upper epidermis stomata is absent, sir, means number will be there, but suppose when compared to the lower epidermis, no, in upper epidermis kammi number irukku. So next one, sometimes here one more important point, sometimes upper epidermis means adaxial epidermis may not consist of stomata. Sometimes absence also we can find out, means sometimes absence of stomata also we can observe on on upper epidermis or adaxial epidermis. So means what is the point now I am going to give you is, suppose if you take upper epidermis and lower epidermis, on upper epidermis we can find out the lower number of stomata than the which one lower epidermis. Why? Because lower epidermis only consists of more number of stomata than the upper epidermis. This is the important point, but easy da. One more point, let me revise once we are talking about the dicot leaf now. So dicot leaf means simply here we are studying stomata is present, more number of stomata is present on the lower epidermis, under epidermis nalli konchu kammi irukku, likewise we are studying. But ipo ulle povumboda, ipo suppose if you are entering into the stomata, no, there God cells are there. What is the shape of the God cells in stomata? What is the shape of the God cells in stomata? No, definitely that is his bean shaped or kidney shaped God cells are present in the stomata. So that particular point you don't forget now. Why? Because here they didn't mention, but I am revising now. So now here don't forget, in stomata God cells are there, two pair means two God cells are there. These two God cells shape is like a bean shaped or kidney shaped. So some lower books they have given as kidney shaped, and in some other books they have given as bean shaped, but shape is important for us. So next one, the tissue between the from here onwards inkonchi important. So from this, so where it is, right, now the tissue between the upper epidermis and lower epidermis. What is the tissue is present? Once you come on to the diagram, this diagram is visible only, no? Once you see this diagram is visible only, no? So in this particular case, so this particular diagram is visible or not, that only you see. So once you see there, so the anatomy of this particular anatomy like a dicot leaf structure, once you observe, no? So now here what is information? Once you see here, like a cuticle is there, and the upper epidermis is there, and here it is a lower epidermis. Now what is the information? What is the tissue? Once you listen carefully, what is the tissue which is present between the upper epidermis and the lower epidermis? This entire information tissue is there, no? One type of cells are there, here one more type of cells are there, no? Arrangement. This rendume together we will be calling as, once you come on to the information, we will be taking as mesophyll tissue. Now you will listen again. The tissue which is present between the upper epidermis and lower epidermis we are calling as mesophyll tissue. Mesophyll tissue. Next one here, mesophyll tissue which possess chloroplast. So means here definitely, so here mesophyll tissue is ellame mesophyll da. If you see here the cells arrangement, no, one type of arrangement is there, here somewhat difference is there, means here these cells and here these cells arrangement is different, no, but ellame together we will be taking as mesophyll tissue. These mesophyll tissues consist of or possess which one? Chloroplast. So when chloroplast is present in that particular cells, no, definitely carry out the process of photosynthesis. And these mesophyll cells are made up of parenchymatous tissue. Simple tissues, our knowledge point of view. So three types of tissues are there, no? Parenchyma, cholenchyma, sclerenchyma, likewise. So we have seen in earlier classes. Now here mesophyll tissue is made up of parenchymatous tissue. Now it has means which one? Mesophyll tissue. Mesophyll tissue is of two types of cells, or it consists of two types of cells. What are those? Now you come here. One is, initially we are calling these two types of cells as one name. That is mesophyll tissue. Now let us learn deeply. This particular mesophyll tissue we will be dividing into two types for our convenience to study. What is the tissue which is present towards the upper epidermis? We will be taking as a palisade parenchyma. And what is the tissue which is present below the palisade parenchyma? Now we have been taken as sponge parenchyma. Now you come on to the information. Now you see, listen again. Mesophyll tissue is there, no ma? This particular mesophyll tissue, now we have been divided into two types, or it consists of two types of cells. One is a palisade parenchyma and one more is sponge parenchyma. Okay, once you listen this, adaxial epidermis. Adaxial means meleva kileva, mele, upper epidermis. So adaxially placed parenchyma or palisade parenchyma is made up of elongated cells which are developed vertically. Once you listen carefully again, means adaxially placed palisade parenchyma what is the tissue is present towards the adaxial epidermis. Author has written like this, how adaxially placed palisade parenchyma is made up of like elongated. Once you see the cells, konjam elongated and konjam enlargement is there in cells. And here these cells you see there, these cells are spherical arc, but here these cells are elongated cells are there. Elongated cells and which are arranged vertically. See there, like honey next will be there, no? Likewise these cells are there, no? So again without intercellular spaces, without intercellular spaces, and the cells arranged in vertically and parallel to the each other. So here the cells are arranged vertically, so here like this vertically and parallel to the each other, so without any intercellular spaces. So here author not given information regarding the presence or absence of intercellular spaces, but my dear students, say one important point in your mind. What is that? In palisade parenchyma, no chance to the presence of intercellular spaces. So next one, once you see remaining information and later information, later oval or rounded loosely arranged sponge parenchyma is situated below the palisade cells. Ipo parenga inge palisade cells it was taken as elongated cells. He has given information regarding palisade cells or elongated cells. Sponge parenchyma point of view, again what is the information we are studying? This sponge shaped or the sponge parenchyma is made up of oval, egg shaped cells we will say, no, adi or round, okay, spherical shaped, and these cells are loosely arranged parenchyma or loosely arranged sponge parenchyma we will observe. And it is situated below the palisade cells. It is where it is there, just below the palisade only, no. So thereby this was sponge parenchyma which is situated below the palisade parenchyma. Now one point you try to discuss differentiation. Now let us see. Palisade cells are vertically arranged and arrangement also is parallel to the each other without any intercellular spaces. Sponge parenchyma, if you see there, no, these cells are arranged in shape is like a motte shaped or like a oval shaped or a round shaped we are taking, and between the cells one more important point we are finding. Once you see exactly at spotlight here, between the cells we are finding a gap, no. So you see there gap is there, no. Particular gap is there means definitely if these cells are loosely arranged only, no, not compactly arranged, loosely arranged cells are there. So that particular thing also we have to differentiate between the palisade parenchyma and one more is sponge parenchyma. My dear students, chinna easy points only but important. Chinna points, no, but important, but very easily we can save in our mind. Okay, next one, there are numerous large spaces and air cavities between these cells, and again continuation here. So here chinna gap is there, no. So here this particular gap here we didn't draw any cell here, and here these cells means exactly these cavities we can find nearer to the stomata. Once you see, so let me show the stomata's structures also. Later I will move forward. Exactly at spotlight and exactly at spotlight and spotlight we are finding somewhat differentiation now, and here also we are finding whether it is appearing or not clearly, but no problem. When you will be here, I'll explain this entire information. Suppose if you have any doubt. Now here once you see there, what is the information? Inge 3 stomata we drawn, upper side 2 da we drawn. So thereby here what is the point we can save in our mind? Lower surface or lower epidermis only consists of more stomata than the upper surface. Ange one neat bit coverage. So now here once you see there, empty spaces are there, no. These empty spaces only author calling as air cavities. So what is the use of these air cavities? Definitely helps in air cavities means air kaati is there, so air will be there, and definitely helps in exchange of gases. Definitely stomata is there means 2 functions it can do. One is transpiration and one more is exchange of gases. exchange of gases point of view, what is the organ is there in the leaf means definitely kandipa we can say one point, sir, stomata, sir. Stomata definitely helps in exchange of gases, and one more is one more, what is that? Exchange of gases, and one more is transpiration. So likewise here empty spaces, where we will be finding of empty spaces, no, these empty spaces we will be considering as air cavities between these cells. Next one, this most important, in 4 lines information is there, no, now only you try to by heart. Actually I should not say by heart, but this is neat bit, that's why if you learn now only without waste of time, no, so to learn this bit no need to concentrate by one more time. So that's why. Now vascular system includes vascular bundles. Vascular bundles we see xylem, the presence of xylem members and phloem members together we are calling as vascular bundles. One tissue conducts the water and minerals, one more tissue conducts the food materials, we see no. So four types also we seen. Did you remember? Adida. So now vascular system includes vascular bundles which can be seen in the veins and midrib. Means the presence of vascular bundles where we can see veins and midrib. If you see, if you take a dicot leaf, no, there midrib is there, and midrib, no, again later divided into divided into somewhat subdivided and arranged like a network like arrangement. So that kind of network like arrangement only, no, our subject point of view we are calling as reticulate venation. Did you remember? So thereby in a dicot leaf or in a dorsiventral leaf, no, so leaves veins arrangement is reticulate venation. After one midrib is there, later that midrib is subdivided into veins and venlets. So we have learned in morphology chapter now. So here what is the point? So midvein is there or midrib is there and veins are there, venlets are there. Okay, adi pakkadalli podinga. Ippu main important point is now the size of the vascular bundles are dependent on the size of the veins. Ippu veins nalla size adhikama irukana, vascular bundles size also will be more. Suppose here vein size is very small, means vascular bundles size will be automatically very small. So thereby once you listen this, try to understand the size of the vascular bundles are dependent on the size of the veins. One more time, the size of the vascular bundles are dependent on the size of the veins. Veins size perusarkana, vascular bundles size also perusavu. Suppose here the vein size is coming avana, so vascular bundles size also will be very small. So this particular point, so while watching video also pass panni, so once you check yourself, suppose when you will be knowing about this one, then only try to move forward to learn the another information. Next one, once you see, so here what is the information? So here the veins. Now you see the veins vary in thickness in the reticulate venation. So here if you see this one, no, the thickness of the veins also changes. Vary means change only, not difference. Vary means, okay, new word. Suppose if you don't know, you can learn this. Vary means difference. So veins vary in thickness in reticulate venation. Appadena why here author mentioned reticulate venation? We are discussing dicot leaf, no, that's why. Next one, now here see there vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. One more time you see there, vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Means here if you see this, no, so there definitely what are the vascular bundles are there? All these vascular bundles, all these vascular bundles are covered by bundle sheath cells. Now diagram paranga. Once you see this diagram, so here we have drawn a pink color. This is color? This pink color is there, no? Outer layer. This is bundle sheath cell. So that particular bundle sheath cell, so now one more neat bit I will give you. This particular bundle sheath cell, initially it is made up of parenchymatous. This bit not there in our textbook. So this particular bundle sheath cell is made up of parenchymatous initially. After first time, apro this later transformed into cholenchymatous. Okay, what is the bundle sheath cell is present there. So this particular bundle sheath cell initially it is made up of parenchymatous, later it is developed into or transformed into cholenchymatous. Next one, if you see this inner side, no, one particular point, my dear students, you try to remember, like a stem we see, no, in stem, no, upper side we have drawn a phloem, and the lower side we have drawn which one, xylem. Okay, means here the xylem and phloem members information point of view. So like that we have learned. But in leaf, no, arrangement of xylem and phloem members is reverse. Reverse means sir means xylem is situated towards the upper epidermis and phloem is situated towards the lower epidermis. But if you take vascular bundles in a stem, no, so phloem we have drawn up. Once you see internal structure of dicot stem, there first of all we drawn phloem, later we drawn a cambium, upper we drawn xylem members. Emma, did you remember the diagram? Or otherwise, once you see the diagram from the textbook, suppose if you have with you, or Amma, do one thing. So here one point I will tell you. Neet, our textbook points. So suppose if you don't know how to purchase, go to try to download one app, NCERT books. NCERT books are there. You will find you can download our textbooks. NCERT textbooks they have given. So NCERT books app only. So if you go there, no, there biology after download, no, biology click paninga, 11th click paninga, apron lesson if you download means there we can find out. Don't worry, suppose sir we are not having textbooks, sir how we have to find out all these means, no worry, download the app, so you will have textbooks virtually. Now here you see there phloem members and here it is xylem members. But here between the xylem and phloem members there is no chance to the presence of cambium. So here cambium is absent. So thereby here suppose when this one means between the xylem and phloem, here I am exactly at spotlight, between the xylem and phloem there is no presence of cambium. So here thereby cambium is absent. So thereby here also conjoint collateral closed type. Closed only, no. Suppose here cambium is absent between the xylem and phloem members means, so what we have to call closed type. So try to remember. So important points, romba important points are there, but if we have interest to learn, no, definitely we can learn. Sarema, so listen carefully. Next one, let us start to discuss one more information. So here now isobilateral. Next one, isobilateral or monocotyledonous leaf. Isobilateral or monocotyledon or monocot leaf simply. Here I will conclude this one within 5 to 10 minutes. Now you see there isobilateral. First only now let us learn isobilateral. Iso both sides are similar. Emma, don't forget isobilateral. One more time you see there isobilateral. Both sides leaf seems to be same. Rice or wheat or maize plants suppose are there beside to our home. Once you go out and once you take and come, that particular leaf once you observe there, both sides color same marka same ilia once you try to observe. Definitely you will notice, what you will notice, both layers means upper epidermis as well as lower epidermis you will observe with the same color. So that's why here monocotyledonous leaf simply we will be considering as isobilateral leaf. Now anatomy, if you see there, here 4 or 5 points, most important points are there. Let us discuss and let us conclude. Now anatomy of isobilateral leaf is similar to the of the dorsiventral leaf in many ways. Means most of the cases, most monocot leaf as well as a dicot leaf similarities are there, but it shows somewhat characteristic differences. So this particular monocot leaf showing characteristic differences. So what are those? Now let us see. So in an isobilateral leaf means which one? Monocot leaf. Okay, monocot leaf. Stomata are present on both surfaces of the epidermis. So here both on both surfaces of the epidermis also stomata are there and with equal number. Emma means here the stomata are present on both surfaces of the epidermis and in equal number. Once you see here one stomata we drawn, here one more we drawn, and here one, appadena what is the point? Inge rendu irukku in upper epidermis and here lower epidermis nalli rendu irukku, so thereby equal number of stomata only is there, no. But whereas in upper epidermis inge in a dicot stem, no, in upper epidermis rendu we drawn, keele we drawn 3, so means here a differentiation is there, Emma. But here in a monocot leaf like stomata information point of view, so the distribution of stomata is equal on both the surfaces of monocot leaf. Emma, so try to learn that particular point. Ipu paringa next line, no, the mesophyll tissue. Ipu here mesophyll tissue in a dicot leaf we have been divided into two types, no, palisade and one more is a sponge. But in in case of monocotyledons, no, mesophyll tissue is not differentiated into palisade and sponge. There is no any differentiation. Here palisade, here sponge, likewise there is no differentiation. Ellame ore ore tissue da. Emma, so next one in grasses, amma idi important. So most of the times, no, they are asking this neat bit only. Emma, in grasses certain adaxial epidermal cells along with the veins modify themselves into large empty colorless cells. One more time, large empty colorless cells development is there. These cells we will be calling as bulliform cells or motor cells. Emma, so what are the, once you see the diagram, Emma, you will be knowing. So once you see there upper epidermis is there, on the epidermis ipo idu cell koncham perusada irukku. Emma, this particular cell only specialized cell we will be taking as bulliform cells or motor cells. Bulliform cells or motor cells. Sir, ipo specialized cell or empty cell or larger cell or colorless cell we are taking, no, which is present at the upper epidermis. Then what is the use of this one, sir? Appadena ipo paringa next to that is only we are discussing. Ipo paringa, when the bulliform cells in the leaf have absorbed the water, appadena these bulliform cells absorbs the water. When these bulliform cells absorbs the water, no, definitely if we eat our lunch, edo biryani, edo we suppose if you eat means generally stomach will be filled. When we will be observing in with empty stomach and filled stomach differences, no, like fully like stomach, suppose if the stomach fully filled means at the time, no, konja it will come out. Now suppose with the empty stomach means adi ulleda irukka. So likewise here also what is the thing, amma adi example only ra, again don't take it in another way. Next one, likewise suppose if the bulliform cells absorbs the water at the time, no, that will be bulged. Our subject point of view only we will be taking as turgid. Turgid, suppose if the cell is completely filled with water, particular cell shows turgidity nature. Turgid cell and the leaf surface is exposed. Appadena when you see that these particular bulliform cells are there at upper epidermis, no, so because of the presence of bulliform cells, so that particular leaf is exposed. Appadena rolling and unrolling processes are likewise two processes are there. So here because of the when water molecules are entered into the bulliform cells, no, leaf surface is exposed. And when they are flaccid means tanni ellame veli varum. So from these cells to when ellame water veli varum, no, means move out, no. So due to the water stress conditions, water stress conditions means due to water lagging conditions, so water tanni availability not there means at the time, no, they make a leaf curl. Appadena at the time in absence of water or during water stress conditions, leaf curl. So leaf curl inwards. Appadena ulleke fold down. So thereby why, sir, like that it is a curling towards the inside means to minimize the water loss. I hope my dear students you understood this particular point. Appadena when particular plants are having of more water, the leaf surface is exposed. Suppose if the leaf not consisting of much water at the time, no, so leaf rolling, that is also leaf curling also towards the inside. Why, sir, like that means to minimize the water loss. Next one, the parallel venation is so this is monocot leaf, no, so thereby here we can see the presence of parallel venation in monocot leaf is reflected in the near which one, similar sized vascular bundle. So once you see that konchum like LMA same irukku, but here one differentiation is there except main vein. So this main vein konchum perusuda we have drawn, but rendu me pakkathale rendu irukudana idu konchum chinnada irukku. Once you see konchum da chinnadu, so thereby here what is the point, in monocot leaf is reflected in the near similar size of vascular bundles except in main veins as seen in vertical sections of the leaf. Once we have seen, no, so there so here upper epidermis is there, lower epidermis is there, between this one tissue is there. Next one, what is the tissue we will be taking, just a mesophyll tissue only. Next one, here these vascular bundles are surrounded by bundle sheath cell is there, no ma, the bundle sheath cell is made up of sclerenchymatous. Don't forget ma. So bundle sheath cell of dicot leaf, no, here it is made up of initially parenchymatous, later cholenchymatous, but here in monocot leaf, no, bundle sheath cells are made up of with sclerenchymatous. Next one, once you see there, this is phloem, exactly at spotlight amma, konja green color irukudana, this is phloem, upper side no phloem, sorry, xylem. So ipo xylem is pink color, green color phloem. Between these two there is no cambium. So cambium is absent appadana. So these vascular bundles are also closed type da. So here the bundle sheath cell is made up of sclerenchyma, green color structure phloem, and blue pink color no xylem. Okay, between xylem and phloem there is no cambium, that's why closed type. And here also we can find out which one empty spaces, no, empty spaces we will be taking as substomatal cavity. So substomatal cavity, like how we have seen in air cavities, no, function is the same, exchange of gases. So dumbbell shaped. So here the what are the cells are there, or the God cells, no, God cells shape is dumbbell shaped. Dumbbell shaped stomata is present. This is a somewhat differentiation between the dicot leaf and monocot leaf, my dear students. So one request, so this much of struggle we are facing to explain this one. So my dear students, watch videos without skip. Suppose if you watch means you will learn the knowledge, so thereby definitely in your life you will reach a good position. That is only our teacher intention, other than that nothing is there. So listen carefully and try to develop the subject. So likewise if you concentrate on all subjects, no, in after 2 years, no, so without a Neet coaching definitely you can get a Neet rank, so you can go to the medical college easily. So be safe and prepare well and learn the concept. Thank you.